My name is Sonal Navadia and today my topic is pathogenicity of influenza virus. Abstract: influenza is an infectious respiratory disease in humans. It is caused by influenza A and influenza B. Symptoms associated with influenza virus infection vary from a mild respiratory disease confined to the upper respiratory tract and characterized by fever, sore throat, runny nose, cough, headache, etc., affecting heart, central nervous system, and other organ system. In 2009, the first influenza virus pandemic of the century was caused by a novel H1N1 influenza A virus resistant that was previously circulating in pigs. Vaccines were not available on the content of first waves of infection. Current influenza antiviral advantage drugs that target that viral M2 join channel and inhibitors of an enzyme have limitations, as viral resistance against M2 inhibitors is prevalent in the currently circulating human influenza A H1N1 global strains. Virus is resistant to the NA inhibitor oseltamivir has been prevalent among influenza A H1N1 strains. Since just before the 2009 pandemic, the admantance-resistant oseltamivir-sensitive 2009 pandemic influenza A H1N1 viruses replaced the previously circulating oseltamivir-resistant admantance-sensitive seasonal influenza A H1N1 virus that circulated globally in 2008 to 2009. This was the history of influenza virus. Influenza in human. The major burden of disease in human is caused by seasonal epidemics of influenza A and influenza B virus, with most of the infections occurring in children. Although most of the severe cases involve very young or elderly individuals. Seasonal influenza A H1N1 and H3N2 currently circulating in humans, but influenza A H2N2 virus were the only human circulating influenza A virus from 1957 to 1968. Zoonotic human infections with influenza A virus strains of swine and avian origin and rare with specific strain, such as avian H5N1 and H7 and N9 and so-called swine H3N2 variant viruses, being the most prevalent but confined to a limited number of human cases in specific geographical areas with only limited human to human transmission. Influenza in human is a major burden of disease. Influenza in animals. Infection of waterfowl with low pathogenic avian influenza A virus, called as such owing to their lack of lethality in poultry, process little or no clinical symptoms. However, upon a transmission to poultry, LPAI can cause substitutional disease symptoms. In addition, virus with H5 and H7 subtypes can become highly pathogenic in poultry. Influenza A virus can also cause mild to severe respiratory infection in a variety of mammals, including swine, horse, and dogs. Additionally, an outbreak of until influenza A virus of avian origin was detected in cats in New York, at first reported in December 2016. From its first report until February 2017, the influenza A H7N2 feline influenza A virus infected 500 shelter cats and a veterinarian who treated one of the infected cats. Human mechanism. Human influenza virus are transmitted through the respiratory route, whereas avian influenza virus in wild birds are transmitted through the fecal, oral, or fecal respiratory route, depending on the route of transmission. The virus targets epithelial cells of the respiratory or intestinal tract for infection and productive replication. In addition, some avian influenza A virus, especially those of H7 subtype, have been associated with human infections of the eye and conjunctivitis. Severity of infection in human is associated with the replication of the virus in the lower respiratory tract, which is accompanied by severe inflammation owing to immune cell infiltration. Cell entry and viral replication. The cellular cycle of the virus starts with the binding to the target cells. This binding is mediated by the viral H1 and it is also responsible for the hemagglutination caused by virus particles when incubated with red blood cells. HAS from human influenza virus bind perfectly to the salicylic acid linked by an alpha 2 6 linkage to the rest of the oligosaccharide, whereas those from most of the avian influenza viruses favor binding to alpha 2 3 linked salicylic acid. As these bonds are the most abundant linkage in the epithelial cells of the human upper respiratory tract and of the avian intestinal tract respectively. After binding, the virus is internalized in an endosome and the endosome is transposed and acidified, which trigger a conformational change in the viral HA that induce the fusion of viral envelope with that of the endosome. As the endosomal pH varies between host species, the pH stability of HA is one of the determinants of viral tropism. This is image of emergence of influenza A virus from aquatic wild bird reservoirs. Antigenic drift. Influenza viruses are capable of evading the antibody-mediated immunity induced during previous infections of our vaccines by gradually accumulating mutations in HA and NFI. This process known as antigenic drift. This method to quantify antigenic drift of influenza virus strain along with site-directed mutagenicity studies have revealed that substitutional antigenic drift can be caused by one or very few amino acid substitutions adjacent to the functional site in the head of the H1 protein. The antigenic evolution of influenza virus was shown to be more rapid in human influenza virus than in swine and equine viruses, which is presumably related to the size, mixing and age structure of the host population. Antigenic shift. In contrast to antigenic shift refer to drastic change in the antigenicity of the H1 HA of circulating influenza A virus. Antigenic shift is associated with influenza A pandemics. In 1977, influenza A H1N1 virus is not seen in humans since the 1957 influenza A H2N2 pandemic start to co-circulated with influenza A H3N2 viruses. The 2009 influenza A H1N1 pandemic was caused by an influenza A H1N1 virus that was antigenically very different from the seasonal influenza H1N1 virus circulating at the time and resulted in the extinction of the previous influenza A H1N1 human linkage, but it did not result in the extinction of the influenza A H3N2 viruses. Immune response. Host innate immunity represents a circular barrier that viruses need to overcome to replicate and propagate in new host. Influenza viruses dedicate several viral proteins to overcome this response. Influenza virus life cycle. Influenza virus enter the cell by endosomal uptake and release. After this cell is entry in the virus, and after the cell is released from the endosomal, and after the cells are enter in nuclear import. In this, viral mRNA is translated into the viral proteins in the cytoplasm, and these are assembled into new virions together with newly synthesized VRNPs. PB1F2 is shown here as a dimer but can also be multimeric. HA hemagglutinin, MA matrix protein, M2 membrane protein, NA neuraminidase, NEP nuclear export protein, NP nucleoprotein, NS1 non-structural protein, PB1, PB2, and PA viral RNA polymerase are present in this virus life cycle. In this phase, how influenza virus enter the cell and which type of proteins are present in the virus life cycle, which is shown in figure, is present in this phase. Diagnosis. A diagnosis of influenza is made mostly on the basis of clinical presentation and epidemiological likelihood of infection. Initially, non-specific symptoms predominant, including fever, chills or frank shaking, headache, muscle pain, discomfort, or anorexia. The onset of these symptoms is sudden, and respiratory symptoms, particularly a dry cough, sore or dry throat, and nasal obstruction and discharge are usually also present. Cough is most frequent respiratory symptoms and can be accompanied by substantial discomfort or burning. Vaccine manufacture. For vaccine manufacturing, first we take circulating strain and master highly growth strain. We mix these both of strains and inject in embryogenic egg. From this embryogenic egg, we select some of strains and amplified on the egg. After amplification, we harvest this strain and particularly purify this strain, and after this purification, inactivated influenza vaccine is manufactured. An antigenically representative circulating strain is restored with the high growth influenza A Puerto Rico 8 1934 virus strain by co-infections in eggs, and a vaccine virus is selected with the highly growth properties of PR8 and the hemagglutinin and NA derived from the circulating strain. The vaccine virus is amplified in eggs, and the allonetic fluid from the infected eggs containing highly titers of the virus is harvested. The virus is inactivated, treated for splitting of individual virus component, and subjected to particle purification to enrich for the HA viral component in the final injectable vaccine. Vaccine. After the causative agent of influenza was identified to be a virus in the 1930s, attempts were made during World War II to develop inactivated virus as a vaccine, which culminated in the licensing of the first influenza virus vaccine for the civilian population in 1945 in the United States. In 1947, seasonal epidemic strain had changed sufficiently that was vaccine made with earlier circulating strains had lost their effectiveness. All currently available influenza virus vaccines are injected with the expectations of the live attendant influenza virus vaccine, which are administered intranasally. Antivirals. Together with vaccines, antiviral drugs play a vital part in the prevention and treatment of influenza virus infections and disease. In a normal influenza season, antiviral drugs are primarily used for treating patients who are severely ill, particularly those who have compromised immune system. In a pandemic settings, especially in the period before a vaccine is available, antiviral drugs are essential for both treating patients who have been infected and preventing infections in individuals who have been exposed. Two classes of drugs are currently approved for influenza, namely the AID and NA inhibitors. Resistance. The emergence of drug resistant virus is a major challenge for the antiviral field. Admenintense resistance first emerged in influenza A H3 and 2 viruses in 2003 and became prevalent worldwide by 2008. The pandemic 2009 influenza A H1N1 virus, which is presently circulating as a seasonal H1N1 virus, is also resistant to the admenintense as its mRNA segment was inherited from a virus that carried the S3A1 mutation 180. Human influenza. People with clinical symptoms of influenza virus infection should remain home and avoid contact with non-infected people until symptoms are alleviated. As the level of virus replication is corrected with the clinical severity of illness, strategies to limit viral replication with antiviral agents would be expected to be effective in acute influenza illness. Animal influenza. As influenza in wild and domestic waterfowl is primarily spread through contaminated water treatment, biosecurity and indoor raising of animals are the main strategies to prevent transmission. Poultry vaccines are inactivated whole virus in oil emulsion, recombinant DNA or vectorized vaccine. These vaccines can be highly effective at reducing disease signs and interspecies spread, but they do not induce sterilizing immunity and have not released an irradiation from endemic countries. Quality of life. Uncomplicated influenza in otherwise healthy adults and children is rarely fatal but can be highly disruptive and lead to substantial absence from work or school. Recovery can be prolonged with persistence, fatigue, and malaise that can last for several weeks after recovery from the other symptoms. Influenza is associated with decreased job performance in adults and reduced level of independent functioning in older adults. Primary influenza viral pneumonia was well documented in the 1957 to 1958 pandemic, although many deaths of young healthy adults in the 1980 and 1919. Thank you for attention.